last part because it was a bit rushed, so I'm going to do the last calculation again. But any questions before that? Yes?
We were studying the mutations which adapted against the bacteria. Do these mutations also can occur against the antibiotics as well?
Same story for antibiotics. So the experiment can be done not with a virus at the end, but with antibiotics at the end, and the same story will happen.
So will it mean like if I don't expose the bacteria to antibiotics, it can still adapt against it?
So again, adapt is the wrong conclusion. What we learned from the experiment is that I grow the bacteria, and then I expose them to antibiotic, and it's not that they see the antibiotic and they say, okay, I need to do this and this. The mutations pre-exist, random mutations, happen to be in the genome, and some lucky bacteria survive, and it's absolutely based on chance. Okay? So again, if you do the same Luria-Delbruck experiment for antibiotic at the end, you will see the same effect. The variance is much larger than the mean. It's random mutations that drive evolution rather than being exposed to the antibiotic and then trying to adapt. Okay? So we had two hypotheses. The Lamarckian one, it was easy to rule it out because it predicted a variance which is equal to the mean. We understood that's a limit of the binomial distribution, which is the Poisson distribution, which plays a major role here. And then we looked at the second model, where you have your growing tree. You're only exposed to the virus at the very end. So we had G equals 20 generation. We started with about 100 cells. It's not very important. You can think about it this as 1. You end up with about 10 to the 8 cells, which I called N, or sometimes NF for final. Doesn't matter. And only at the end, you expose the bacteria, these bacteria, to the virus, and you count how many survivors. So the output of every experiment, Sunday you get 0 survivors, Monday you get 15, and so on, and you make a histogram. And we saw that it has this power law tail, approximately. We're going to talk about it. And the variance to mean was very large, hundreds, much, much larger than 1. And this is what we're trying to understand. And I'm going to repeat the kind of last calculation slightly more slowly. So what we realize is that it's convenient to split things according to the generation. It's kind of a natural way to split the data. And we realize that the mutation events, they're completely independent. So if I look at the number of mutations that happened at each one of the layers, let's say I look at the 7th layer. Okay, so I have 2 to the 7 cells. And if I ask you, what is the number of mutations, not survivors arising from them, but the number of distinct mutations that happened on that generation, what's the form of this distribution going to be? Poisson. Okay, so again, even though now we're talking about the right model, Poisson distribution is still playing an important role in the calculation. So I know that if I look at the number of mutations in the nth generation, it's going to be n0 times 2 to the n times some p. Okay, that's the average number, or that's the average number of mutations arising in the nth generation. And if you ask me what's the entire distribution, I know that it's going to be the Poisson. Is that clear? Okay. So what's the difference between Mn, the number of mutations? If I think about Mn as the random variable corresponding to the random mutations occurring in the nth generation, and we have the Sn we introduced yesterday. Sn was the number of survivors arising from mutations occurring in the nth generation. It's almost the same thing. What's the relation between the random variable, this is a random variable Sn, and the random variable Mn? What is the separating them? Which factor? 2 to the g minus n. Okay? So this is a constant factor of amplification. If I got a mutation here, all of these are becoming all of these cells at the end will become resistant. So I get amplification by this constant factor of 2 to the g minus n. It's constant, but it depends on the generation. If I got the mutation early, I have a huge progeny that are all resistant to the virus. But now it makes my calculations very easy because Mn is Poisson, right? So I know that the mean is equal to this. And that tells me that the mean of Sn, we saw it yesterday, is simply n0 2 to the np times 2 to the g minus n. And then we had this nice result that this is simply 2 to the g times n0, which is the n final times p. So this was the surprising result that the contribution from every layer is equal. The third layer has a very small chance of having mutations, but big effect. So on average, if I average over a huge number of experiments, it contributes the same as the 7th layer or 8th layer or 9th layer. And that gave us the result that the total number of survivors, this is a random variable that is approximately equal to the sum of Sn. We realize that the different layers are approximately independent of each other as random variables. And that's why this S average was simply np times g. We had this result yesterday. Okay? But now what about the variance? We need to calculate the variance of S. These are approximately independent random variables. So we can say that the variance of S is approximately the sum of the variance of Sn. Now, let's calculate the variance of Sn. Sn is a constant factor times Mn. Mn, what kind of random variable? Poisson. So what do we know about the variance of Mn? Equals to the mean of Mn. Okay? So now it's very easy. We know that the variance of Sn is simply equal to the variance of Nm times what? And don't tell me 2 to the g minus n. So Sn is Mn times this. Mn is Poisson. So I know that the variance of Mn is actually equal to the mean of Mn, which we just calculated. It's written here. Where is it written? Up. Okay, so it's written here. So we know that. But I'm saying the variance of Sn is not equal to the variance of Mn because it's equal, as a random variable, it's Mn times this factor. So what is the variance of Sn? No, squared. Very good. So this is important. Without this, we wouldn't have any of the interesting results. Okay? So we get this 2 to the g minus n squared, and this is really important. And now we're done. Okay? Because now we see that now it's just simple algebra. Okay? Because we see that this, I can plug in the expectation value of Mn. If I had just one factor 2 to the g minus n, it would be just like the result for the mean. Okay? So it's equal to n times p. But because we have this extra factor, it's n times p times 2 to the g minus n. So now we have, this is the variance associated with mutations from the nth generation. And the total variance of S is simply equal to sum over all the generations, n times p times 2 to the g minus n. So now, which generations contribute the most to the variance? For the mean, it was equal contribution from all the layers. What about the variance? The first generations, right? The first generation are super rare, and they contribute the most to the variance. Okay, now it's very easy to sum up this series and find that the variance of S divided by the mean of S is approximately equal to 2 to the g over g, which is a huge factor if g is equal to 20. It's much, much larger than 1. So that was the main insight, explaining why in this model the variance is much larger than the mean. So you can see the Poisson entered here also in a kind of important way. Okay, and the whole important thing is this amplification by this factor. If this was not here, it would just be a sum of Poissons, very boring. Okay, this amplification is what gives you this huge variance. Any questions? Okay. So that was the first point I wanted to make. One technical point, some of you I see are using the uploaded notes. I keep updating them, so if you're working on them, check if there is a kind of new version. So for example, here this morning I thought about simplifying the calculation a bit, so I want to reflect that in the notes, and I'll probably do that today, and there will be a new version. Okay? I also uploaded this morning, for those of you who want to kind of challenge yourself, 2 problems. They're not mandatory, do them if you like. One is about Luria-Delbruck, and the other is about what I'm going to be teaching in the rest of today, also about evolution. So they involve some simulation, some analytics. If you want to try, feel free. Definitely not mandatory. Okay, so that was a second technical point. One last comment about this. We said that in the actual experiment, you're not going to get this variance to mean because you're very unlikely to sample scenarios where the mutation occurred in the first or second generation or third generation. Okay? So really, when you do these calculations, you need to replace these sums of n going from 1 to g, of n going from some g effective to g, where g effective is the first generation at which you start to have a real chance of seeing the mutation. And we said that the criterion for that is very simple. It's simply 2 to the g effective times n naught. That's the number of cells I have in the let's say, 9th generation, if it turns out to be 9th. That times the probability times the number of experiments should be of order unity. This is a criterion for saying if I did 300 experiments, what is the first generation at which I have a reasonable chance of seeing a mutation? And so if you kind of use this as the definition of G effective, plug it into these formulas, you will get a modified variance to mean that is going to be much larger than 1, but much smaller than this. And that agreed well with the experimental results. They already did that in the original paper. It's a kind of technical point. It's not super interesting. So if it's unclear, and it's quite heuristic, so you can, if you don't like it, an alternative is to run the code we discussed yesterday for not 10,000 experiments, but 300 experiments, and see empirically what's the variance to mean you will get. You will not get this value. Again, because of this effect, you're not sampling over the rare cases of getting mutations very, very early on in the tree. Is that point clear?
But in the rest of the that in the exponential you have minus GF, in the
Exactly. It's very simple to modify. Yeah, absolutely. Okay. Other questions? Comments? Okay, I'm going to say a few more words about Luria-Delbruck, and then we're going to move on to discussing the implications of random mutations on the course of evolution and talk about some additional evolution experiments. So one thing I wanted to mention is that somebody hinted at that yesterday. When you take this Luria-Delbruck experiment in the case of a very large number of generations, so it's not we're kind of diverging away slightly from the original experiment. There are lots of mathematical works inspired by this original experiment. And one of them is asking what would be the distribution in the limit of many generations, if it wasn't 20, but 100 generations. And in that case, the distribution turns out to have this form. Okay? It increases and then decreases and decays as a power law with a power of 2, 1 over x squared. And it was realized by Mandelbrot that you might have heard about from fractals and things like that, that the Luria-Delbruck in this limit generates a very famous function, which is called the Levy-stable distribution. Okay? Levy-stable distributions are the generalizations of Gaussians in the case of, so it's a kind of fate, what happens to the central limit theorem where your distribution doesn't have a finite variance. Okay? In order to get a Gaussian limiting case, you need a finite variance. If the distribution has a power law tail, there is still a theorem about the limiting distribution you converge to, but it's not Gaussian. It's a family of functions called Levy-stable distributions. If the power law you start with is 1 over x squared, you converge to this particular function, which is essentially a Levy stable distribution, but this one has another name. It's also called the Landau distribution because Lev Landau bumped into it in the context of a completely different physics problem involving particles moving through some neutral gas that takes their energy, and you ask what's the energy distribution. So nothing to do with evolution, but you get the same math and the same. So this is called the Landau distribution. Okay? We can get a bit of a hint of why this has a 1 over x squared distribution if we look at essentially the math we did here on the board. So let me quickly do that. Okay. So if we look at our tree and we say we got a mutation at some generation n, same sort of math. The probability of that, Pn, was, let's take n naught to be 1 for simplicity. So it's 2 to the n times p. That's the probability of getting mutation at generation n. And then it leads to the number of survivors. Let's assume that we're in the case of kind of very small p, so we get at most 1 mutation somewhere in the tree. And then the number of survivors would simply be 2 to the g minus n. Okay, so I got 1 mutation somewhere here, and that's the number of survivors. It's simply this simplification. So that's 2 to the g minus n. Okay, so we have the probability to get the mutation, and we know the impact. So if I want to know what is p of s, what is my probability distribution to get s survivors? Okay, I can equate p of s, and I'm going to view it now as a continuous variable. Okay, so p of s ds, I can say it's p of n dn. Okay, just change of variables associated with the probability distribution. So p of s is equal to 2 to the n times p divided by ds to the n, okay? And ds to the n gives me 2 to the g, let's do it like that. Okay, 2 to the g minus n up to some factor, okay? And this is proportional to 1 over s squared. Okay? So when I do this simple math, I see that the distribution has a 1 over x squared tail, which is supported by the simulations I'm showing you on the right over there. Okay? So this was one small comment, not too important, but it was related to a comment somebody made yesterday about when we said how do we analyze this distribution, maybe we want to fit it to a power law. So indeed it has a power law tail with a specific power of 2. One last comment I'm going to say about the Luria-Delbruck. So I told you it inspired a lot of probably tens of follow-up mathematical works because it has this rich mathematical structure. And recently we were motivated by a different experiment to look at what happens in the Luria-Delbruck experiment, but when the mutations are much, much higher. In the original experiment, they were of the order of 10 to the minus 8. We're interested in what happens in the regime where the mutations are of order 1. Why were we interested in that? Because of this experiment. So you have here yeast cells. I showed you yesterday yeast cells growing and dividing. So you have something like that. You have yeast cells growing and dividing, but their DNA is damaged. And in a biological process I won't get into, here their DNA can be repaired by CRISPR. The details are not important. It's not mutations, but mathematically it's a process that happens with some probability. And once it happens, once the DNA is repaired, cells, in this case, turn green because you're making some fluorescent protein functional, and it's heritable. So mathematically, it's equivalent to the Luria-Delbruck experiments, except that the effective mutation rate, it's not a mutation here, it's DNA repair, is of order unity. Okay? So now we're in a kind of completely different regime where all the approximations we made because p was small are not valid, and you have to do everything again. And what we found is that if you look at this experiment, eventually all the cells will turn green because at every generation there is a chance of a cell becoming green, and once it becomes green, it stays green, and all its children become green as well. But if I look at the variability in the number of green cells, it can either increase over time or decrease over time. Why is that? Because I have a competition. Over time, I get many, many, many cells. Okay, so that kind of increases the variance. But on the other hand, the fraction of green cells approaches 1. So that reduces the variability. Ultimately, everything should become green. So you have a competition between these two effects, and we found that there's a critical probability of half above which the variance decreases exponentially with time, and below which it increases exponentially with time. And now you can use this result to look at the experimental data and infer information about the DNA repair process, in this case associated with CRISPR. So it's in the spirit of Luria-Delbruck in the sense that we're relying on the variability in the experiments to learn something about the biology, and this theme will come again in the second topic we'll discuss probably tomorrow, of cell size control, where we're going to learn from natural fluctuations. Okay, so that's, I think, the takeaways from Luria-Delbruck. If you've got variability, sometimes you can learn from it, and it's not just a bad experiment. It's actually the information is the variability between the experiments. It's sometimes called the Luria-Delbruck fluctuations test. That's another name for it, so you can learn from the fluctuations. 2, we saw that you can have a physical model without forces and without Maxwell's equation, and it's still very much the sort of calculation that physicists do. Okay, so physics is beyond the standard thing you learn in undergrad. It's much broader than that. And I think we can kind of now talk about the implications of this. So I'll pause for questions, and then we'll stick with evolution, but talk about implications of random mutations on the course of evolution. Any final questions on this? Yes? Yes. So your question is, okay, your question is why would they keep it and not lose it? Okay, so that's maybe it'll become clearer. Wait until the end of class, because there is something very artificial about, I mean, it's an experiment. We can do whatever experiments we want. So it was a useful experiment. They got a Nobel Prize for it, and they deserved it. But there is some feature of this experiment that is not unlike the real world. What do you think in this experiment doesn't mimic the real world? They're not limited by resources. They're growing exponentially, and we are keeping everybody. Now, if you do a simple calculation, take E. coli cells, let them double in 20 minutes. In 48 hours, their mass is larger than the mass of planet Earth. Okay, so good luck with keeping things exponential in the real world. Okay, so in the real world, a different model that is the model we're going to discuss today is called the Moran process. Okay, and in the Moran process, you have a box, and in the box you have the bacteria. And let's say you have 10 to the 8 of them. It's not very important. And every time a cell divides, you kick out another cell from the media, right? Or you kill it. It doesn't matter. So you maintain the number of bacteria constant throughout your experiment. Okay? And that's a more realistic scenario for how things work in the world, where you have limiting resources, so fixed population size. And in that case, even if you got say a mutation that gives you resistance to a virus, it doesn't mean that it's going to stay there. Maybe at some later point it will be kicked out. And it's exactly the question we're going to ask, is if I give you a mutation and I wait for a very long time, what is the chance that that mutation overtook the population? Okay? So in Luria-Delbruck, everything was neutral. What does neutral mean? It means the mutation didn't affect the fitness of the population. It has the same growth rate. We assume that the growth rates are independent of whether I got the mutation or not. So if I have this box and I got one cell that I color in red, it has this mutation, and let's say it doesn't affect the fitness. And now I let the cells, and maybe the red cell becomes 2 red cells and 3 red cells, and then it goes down to 2 because I kicked one of them. So it does some kind of fluctuations, the number of red cells. And eventually, either everything becomes red or the red is lost, right? So if I ask you what's the chance that this red cell, out of a population of 10 to the 8 cells, what is the chance that it overtakes the entire population? We're going to calculate this formally, but you can already know the answer now. So what do you think the answer is in this case? It's almost 0, but what if I want to be precise, because this is going to be important? Perfect, exactly. There is nothing special about this cell because we said it's symmetric, right? It doesn't have. So if I color one cell red and another green and another blue, they're all the same. The chance of each one of them overtaking the population is 1 over the population size. And this is called neutral evolution. So when do I have a chance of beating that? Because 1 over 10 to the 8 is really small chance. Well, what if this red cell is doubling faster than the other ones? Then there is a chance that a real chance that it overtakes the population, and that's what we're going to calculate next. So your question is kind of a perfect transition to the next topic we're going to discuss, which is fixation and the course fitness trajectories and the course of evolution. Any other questions? Yes. Fix.
Help you survive or fix something, just like it might increase the rate of growth?
So I'm saying, even if I'm saying, we're going to do this thoroughly, but I'm saying if you have neutral mutation, meaning it doesn't affect the growth rate, the chance of it fixing, meaning overtaking the population, is very, very small, 1 over the population size. And I'm saying even if it has 20% advantage, we're going to see it doesn't mean that it's still going to necessarily overtake the population. Why? Because of stochasticity. So in evolution, stochasticity is critical. Okay? Why do you think in evolution stochasticity is so important? What is the key feature in evolution that inherently makes stochasticity important? Mutations happen at 1 copy. When I got a mutation, by definition, it's in 1 cell. If I'm 1 cell out of 10 to the 8 cells, stochastic fluctuations are going to be important. We're going to see this formally. But that's the reason, because you don't get, if I had 100 mutations happening at once, I can take the continuum limit, stochasticity will not be important. But because of mutations happening at 1 copy out of the entire population, stochasticity is critical here. Okay? So that's the next topic we're going to discuss, evolution, fixation, evolutionary trajectories. Sorry, this is the story I told you about this variance, either increasing or decreasing in this Luria-Delbruck limit with high effective mutation rate. So now we understood that random mutations drive evolution, and we're going to discuss evolutionary dynamics. And this is from a paper by Lenski and Travasino. And they say, imagine you've discovered a well-preserved and clearly stratified fossil bed that provides a record of evolution extending thousands of generations for the particular organisms that you study. Okay? And the fantasy continues. Imagine that you can resurrect these organisms, not just bits of DNA, but the entire living organisms, and reconstruct their environment exactly as it was during the thousands of generations preserved in the fossil bed. Okay? And he continues, yet this fantasy is not fiction. In fact, we have many such fossil beds preserved, and we have traveled in time to manipulate population with respect to their histories and environments. The fossil beds are preserved in a freezer and contain population of the bacteria E. coli. Okay? Our time travel thus far extends over 5 years. This is an old paper, representing 10,000 generations in the system. Okay? And he says our approach might be called experimental paleontology. Okay, so he's not dealing with dinosaurs, he's dealing with E. coli. This experiment started in 1988. Okay, so now it's not 5 years, but more than 30 years. And what he's doing is basically every day growing bacteria, just like in the Luria-Delbruck experiment. So they start with about 10 to the 6 cells every day. They grow them to about 10 to the 8 cells. So the bacteria grow in population to about 100-fold. Okay? And then, so this part is exponential. So they grow exponentially. They run out of food. So now real life comes in. They run out of food after about 6 generations. So G is about 6. And then the next day, they take 1% of their culture and put it into fresh medium. So it's kind of a mini Luria-Delbruck experiment, but that runs out of food, and every day you put it into new media, and they've been doing it for about 75,000 generations from the year 1988. Okay, probably before most of you were born. Yes? So this is the medium, and every day I take like, this is the bacteria from the previous day. This is fresh food. I take 1% from here, and I put it into here. They grow 100-fold, finish the food here, and I repeat it every day, again and again and again.
But for 18 hours, yeah, we don't change because they finish that.
So the doubling time is not 20 minutes. It's slower than that. Okay, so they spend, I don't know, a couple of hours growing, and then most of the day just, finished food and just hanging around. Okay, so that's the experiment. And what are the advantages of this experiment? Lenski was quite a pioneer in kind of foreseeing that this experiment is going to be so useful for understanding evolution. And some of the advantages of using E. coli rather than, I don't know, rabbits or whatever, is that you have a fast doubling time. Okay? Even if it's not 20 minutes in this media, it's still fast. You can do 6 generations every day. You can replay the tape of life. So I think this is a quote by Levontin, another influential evolutionary biologist. What does it mean to replay the tape of life? What can I do with E. coli that I cannot do with higher organisms?
You can see them grow and then put them in different situations and then see them grow again.
But what does replaying the tape of life mean?
You can see the whole thing.
You can freeze them. Okay? You can put them in the freezer. So I can do stuff like compete the cells of today with the cells of 1992. I can literally go to the freezer, take cells from 1992, and make a competition between the cells of today and their great ancestors. Okay, so that's replaying the tape of life. I can do the experiment again. He did another smart thing, which is to use, I think, 6 replicates of the experiment. So he did many runs of evolution in parallel, so you can see how reproducible things are in evolution. What is the role of chance as opposed to, are there convergence, things like that. And the physiology is much better understood. Like we said, E. coli is the best understood organism on this planet. We understand the properties of E. coli growth much better than we understand the mammalian cell growth or tissues. Okay, so things are a bit simpler. Still very complicated, but simpler than higher organisms. And when you look at the results from about, 30 years, you see that this is the fitness. I'm not going to get into how he defined fitness, but it's approximately a competition assay with the original ancestors. So you check how much better you are at growing in this device compared to your ancestors. And you find that the fitness keeps increasing even after tens of years. Okay, so you might have expected that bacteria will quickly increase and then plateau because, okay, that's as good as you can be in this medium, but they keep getting better and better and better, albeit at a slower rate. So kind of it somewhat decreases in the rate at which it happens. And this is well fit by a power law with a very small exponent. So it's approximately a logarithmic trajectory of increase with time. So it increases, but at an ever-slowing rate. And I hope we get to discuss, what that tells us about the possible models. Okay, so that's the Lenski experiment. Yes. No, so it's something that every microbiologist does all the time. You take your strains, you freeze them, and then you unfreeze them and let them adapt to the medium, and they're happy to go. It doesn't damage their DNA. So So the answer is no. It's very conventional. Other questions? Yes. What's happening in? Oh, I think this is Rich Lenski at the back there, and I think it's just when you're doing experiments for 30 years, these are, the Petri dishes, you accumulate a lot of them. Actually, now he kind of, 2 years ago, I think he passed on the baton, and now the experiments are done in a different lab, but it was done in his lab for more than 30 years. Yeah. Okay, so now we wanted to understand evolution and evolutionary trajectories, and the first thing we're going to discuss is exactly so how does evolution progress? Typically what happens is you get a mutation, and if that mutation is beneficial for your growth, there is a chance that it overtakes the population, and you got now a new species of bacteria that grows a bit faster, higher fitness. Okay? But first of all, most mutations are deleterious, meaning they lower your fitness, so they will be purged from the population. We're going to see this in the calculation. And second of all, like I said, even if the mutation is beneficial and helps the population fitness, there is a good chance that it won't fix in the population. And that's the sort of thing we're going to calculate now. Given that a mutation occurred, what is the probability that it overtakes the population? Now, you can do this calculation in the serial dilution. This is called the serial dilution experiment. Every day you grow things and then dilute, and grow things and then dilute. And in the notes that I uploaded, I put a calculation we did a couple of years ago for the fixation probability in this protocol. It's a bit of a more complicated calculation than the one I'm going to do today. So you can look at it if you want, but you don't have to. I'm going to present the qualitative results are going to be very similar, but I'm going to do it in a different model where I don't grow 100-fold and dilute, but do what I said before, fix the population size exactly, such that every time a cell divides, I'm kicking out another cell from the medium. So this is called the Moran process. Okay, and there are other models in evolution, like the Wright-Fisher model. They give essentially the same results. Okay, so different models for the details of how you fix your population size will give qualitatively the same behavior, up to factors of 2 or not so interesting factors. And that's why I chose to do the calculation in the perhaps simplest way, in the Moran process. I'm going to follow a chapter from Martin Novak's book on evolutionary dynamics that I also uploaded to the shared folder. Okay, so that's the calculation we're going to do now. What am I going to say? I was going to ask you, if I look at the serial dilution, where is stochasticity? I grow 100-fold, I dilute. I grow 100-fold, and I dilute. Where is the random point that might make a mutation either fix or not fix? Exactly. The 100-fold dilution, I take 1% of the cell. If I had a mutation happen, I don't know, somewhere in the middle of the day, maybe it didn't have time to reproduce so much, and then there is a chance that when I take 1% of the population, I'm going to not grab one of these mutants. So even if a mutant occurs, there is a chance that it'll be lost. That's where the stochasticity is. Okay? In the Moran process, every time a cell reproduces, I take a random cell and kick it out of the medium. That's where the stochastic element is for that Moran process. So stochasticity is key, like we said, and like we're going to understand now. So now we're going to discuss the Moran process. Okay. So now we have n cells, and every time a cell divides, then we take another cell and get rid of it. That's a description of the process. It would be very easy for you to simulate it and play with it if you want, and that's one of the problems I gave in the problem set I uploaded today. So the first thing I want to do is ignore stochasticity, even though we're going to see that's not going to be possible for the evolutionary dynamics. And let's look at two populations. Let's say I have two populations in my Moran process. I have these cells. I'm going to call their number N1. And I have another species, these guys, the number of which is N2. Okay, and together, N1 plus N2 is exactly equal to N. Now, let's write an ordinary differential equation for dN1 to dt. Okay, so no stochasticity, I'm writing the continuum limit. When do you think this will be justified? Perfect answer. So when N1 and N2 are much larger than 1, I can be in this continuous limit and write this ordinary differential equation. Now we can see why this is invalid for evolution, because like we said, for evolution, you always start with, say, N1 being equal to 1. When you get a mutant, it's always one copy in your population. Then what I'm writing here is not valid. So dN1 to dt is the growth rate, I'm going to call it lambda 1 of this subpopulation N1. Okay? So this is the proliferation rate of these cells N1. But now, as cells are growing and dividing, what is my flux out? How many cells am I kicking out per unit time to account to offset for the growth of the N1 and N2 cells? So my question is, every time a cell divides, I'm kicking another random cell out. So if my N1 cells divide at a rate lambda 1 N1, okay, lambda 1 is the doubling, is the growth rate of the N1 cells. Lambda 2 is the growth rate of the N2 cells. So what is my rate of kicking out cells overall without caring which cells they are? Not just that. The sum. I'm kicking, right? Both happen. I don't, I'm not averaging, right? So the total rate of kicking cells out is lambda 1 N1 plus lambda 2 N2, okay? But I'm not kicking out just N1 cells. I'm kicking out both N1 cells and N2 cells, and I'm choosing them randomly. So what must I multiply this if I'm writing the rate of N1 cells with time? So exactly. So times N1 divided by N. So let me explain this again. This is the proliferation rate of the N1 cells. This is the rate of expelling cells out. And this factor is the fraction of the N1 cells out of the ones I'm expelling out. Notice that here there is no lambda 1 and lambda 2 because I'm choosing the cells I'm expelling completely randomly without regards to their growth rate. Whether you're growing fast or slow doesn't matter, you're kicked out at the same rate. So everybody's okay with this equation? Yes? If not, slow me down. And now, simple algebra will show you that if you look at N1 is simply N1 over N, so that's the fraction, then you find that dN1 to dt is equal, after some simple algebra, to the difference in growth rates times N1 times 1 minus N1. Okay? So what do we find? First of all, does this make sense? What is the behavior of this? Let's say lambda 1 grows 1% faster than lambda 2. Then what will happen when I start with the two populations? Even if I start like with very small percentage of N1, but it grows 1% faster than lambda 2, then what will happen? It will overtake the population. So we don't have stochasticity. So here, no matter how low we started, if we started a very small fraction, if this is time and this is N1, and I started at a very, very low fraction, but it grows 1% faster, then it'll initially grow exponentially, because initially at a low fraction of n1, I can forget about this term, and this is simply exponential growth. So it'll grow exponentially with a rate of lambda 1 minus lambda 2. And then eventually, when n1 reaches high levels, what will happen? It'll saturate at a value of 1. So here, no matter how much better n1 is compared to n2, it'll always overtake the population. Why? Because we neglected stochasticity. By the way, this equation is called, does anybody know what it's called? Logistic growth. Okay. Quite useful in, I'm sure you'll see some of it in the ecology session next week. Okay, so this is logistic equation. Another side comment is, if you look at the discrete version of this, okay, it has nothing to do with what I'm going to discuss next, but I'm just going to mention because it's interesting physics. If you look at the equation, xn plus 1 equals some constant r times xn times 1 minus xn. Looks very similar, right? It's like the discrete version of what I wrote now, but there is a big difference. This is an ordinary differential equation that qualitatively you can see is exponential and then plateau. It's very easy to solve exactly, so I leave it to you as an exercise to solve it exactly. You can find what this form is precisely. You know, standard ordinary differential equations. This, on the other hand, while it looks very similar and simple, is hugely complicated. It's called the logistic map, okay? And it displays chaos for certain values of r. So if you Google logistic map, you will see very, very rich physics and chaotic behavior as a function of the only parameter you change here is r, okay? And it's hugely complicated. And it was very important for the history of understanding chaos and things like that. Not relevant for what we're discussing today. Yes? So you'd think, okay, if I can solve this exactly, I should be able to solve this exactly, but the answer is no. This is much more complicated. It's not one-to-one. Okay, so now we're going beyond this. What we understand is that if I have a population in the Moran process, and let's say I have one cell, let's say everybody is N2, and now I got one N1 cell. This is my mutant. And it grows a bit faster. So lambda 1 is slightly larger than lambda 2. What we understand from this calculation is if I get to some certain level that are high enough for N1, once I get to that point, things will continue essentially deterministically, and it will overtake the population. So all the interesting stuff about fixation is starting here at this one copy number. Here, fluctuations are super important. If you get out of this initial phase and you get here, things will continue and you'll overtake the population. So all the calculation we're going to do now is going to be stochastic, and is going to be essentially to understand what's the chance that I get out of this and overtake the population. So given that a mutant occurred, and given that I have some advantage in my probability, what is the chance that I overtake the population? That's the calculation we're going to do now. It's going to be not so simple, okay? So bear with me. I'm following the book chapter by Martin Novak for this particular calculation. And I'll tell you kind of the result in advance. I don't think it's a super intuitive result. The result is that n, for large N, capital N doesn't matter. And my chance of fixing is proportional to my fitness advantage. So if lambda 1 is larger than lambda 2 by, say, 10%, then up to a proportionality constant that's a bit dependent on the model, let's forget it for a second. If I have a 10% fitness advantage, my chance to fix in the population is about 10%. So if you think about, I don't know, Omicron, and it had a 20% advantage over Delta, and it means that probably it happened 4 times before, and you needed, like, on average, 5 attempts until it overtook the population. So even in a large population, the result turns out to be independent of n, which is sort of surprising. And the fixation probability is proportional to the fitness advantage of the mutant. Okay? And if my fitness advantage is 1%, then I have a chance of 1 to 100 to fix. And if my fitness advantage is negative, I have 0 chance of fixing in a large population. So deleterious mutations cannot fix, and beneficial mutations in a large population fix in proportion to their fitness advantage. That's the result we're going to see now. Any questions? Yes?
Why the proliferation is fixed? What if it is random?
What is random? The proliferation?
The fixed growth.
You mean, you're talking about this?
Yeah, but why each in one population and in two populations has it fixed?
The growth rate?
Yes.
Because the growth rate is an inherent property of the cell. If I take E. coli in a given medium, it grows like a, remember the video we saw yesterday. If I fix the environment, my E. coli cells are growing and dividing at a constant rate.
So if one grows more, he will be the fitness.
But what we see now is because of the random kicking out of the cells, that's where the stochasticity is. Even if one grows more, there is a chance that I will kick it out at an early stage. And once I hit 0, I'm at 0. I cannot come out of from nowhere. So that's the thing. When you're starting with 1, you go to 2, you go to 3, but then there is a chance you go to 0, and then you stay there. So stochasticity is critical, even if the growth is deterministic. Yes?
This question may be a little out of topic, but after 50,000 generation, it is still E. coli. Like, why does it is not a new bacteria?
Well, evolution is very, very slow. So for us, we say 30 years, what a long experiment, how much patience Lenski has. But think about it. These bacteria have been evolving for billions of years. For them, this is nothing. So the question you can ask is, they've been evolving for billions of years. Why the hell can they even do anything for these 30 years? It shouldn't be negligible. So the thing is, now you took bacteria that have adapted very well to a natural environment, life in the real world, and you're putting them suddenly in an environment which is very fixed. So Lenski is giving them a particular medium. Every day they see the same food. Okay? So evolution hasn't prepared them to this particular media. So there is still chance for them to improve over time. But it's very small improvements. Over the course of 30 years, they get a few tens of mutations that are beneficial. They can't do much. So they're not going to become something completely different. They're still very much E. coli, but with small modifications that are suited to the particular medium that they were challenged with. This is the new challenge. OK, so let's start with this calculation. And I'm going to try to follow the notations in the book. And so we look at I as the number there are two types, A and B. Okay, so these are my A and B cells, and I is the number of, say, A. And let's assume to begin with, in the first part of the calculation, that the growth rates are identical for the A and B type. So this is what we call neutral evolution. We're going to do that as a warm-up, and then go to the general case. So for the warm-up, that will still introduce some of the notations, what is the probability to go from I to I minus 1? Okay? So to go at one of the steps in our evolution, in our dynamics, sorry, to go from I to I minus 1, what do I need? I need a B cell to reproduce and an A cell to be kicked out. Okay? So the chance of that for a B cell to reproduce and for an I cell to be kicked out, I get n minus I over n. that's the chance that one of the B cells reproduce, because everybody has an equal chance to reproduce, right? So again, let me define the model more clearly. I have my n cells, and at every step, I choose one cell to reproduce and another cell to be kicked out. Now, later, when we put in a fitness advantage, when I choose the cell to reproduce, I'm going to bias one of the types, the A or the B, okay, to make it non-symmetric in their chance of being selected for reproduction. But now, in the simplest case, I choose one cell for reproduction and another cell to be kicked out. So if everything is equal, I have n minus I B cells. So that's the chance that I selected a B cell for reproduction. So reproduce B. And then I need, in order for the A type to go down by 1, which cell do I need to choose for being kicked out? A type. And what's the chance of that? I over n. Great. So this is my chance of going down. And what do you think is the chance of going up from I to I plus 1? Same, because of the same logic. I need to now choose an A to reproduce and a B to be kicked out. Same story. So I get, n minus I over n squared. And the chance to stay in the same numbers is simply 1 minus pi I plus 1 minus pi I minus 1. So now we've described a Markov chain. If you've never seen Markov chain before, it's not important. This is a Markov chain. I have n discrete, n plus 1 discrete states, the number of A being from 0 until n. These are my possibilities. Either everything is A or nothing is A. So I have n plus 1 states. And these are my probabilities for transitioning between different states. That's all. Okay? And Markov chains are quite useful in general in science and in physics. If you're interested in seeing another example, I'm not going to discuss today, the Google PageRank algorithm is strongly related to properties of Markov chains. You can find it in my book. I have a book on disordered systems and stochastic processes where I talk about Markov chains, and this is one of the examples that I discuss. Okay, so now let's kind of write some other things. What is if I'm at state 0, if I have 0 A's, what is my probability to transition to the 0 state? 1. Okay, if once I'm done, once I have no A cells, I cannot get them out of nowhere. So this is 0, and P of 0 to any other state different than 0, that's 0 because I cannot make them. And if everything is A, then what is P of nn? 1 as well, right? And same story. So now we're almost done. And now I'm going to define xi as the probability of ending in state n starting from I. So what is the thing when we talk about fixation of a mutant that emerged? What state do I start with? 1. And I want to know what is the probability to end up in state n. Once I'm in state n, it's a so-called absorbing state. It's called an absorbing state because once I get there, I stay there forever. So the question is, if I start, really the question we're after is, what is x1? Okay, but like sometimes to solve x1, it's easier to solve for all the xi's for any I. Right? You strengthen what you're trying to prove, and it's an easier thing to prove. Okay? So xi is the probability of, we know that xn, for example, is simply 1. And x1 is really what we're after. That's the fixation probability. And now the trick is, we're going to write a recursive equation for this variable that we're after xi that is going to be related to these transition probabilities of the Markov chain we defined. Okay? How are we going to write this equation for xi? Basically, recursively. And this is very much in the spirit, those who are familiar with Fokker-Planck equations, there is a so-called forward and backwards Kolmogorov equation. So what we're doing now is similar to the backwards Kolmogorov equation, while these transition probabilities are similar to the forward Kolmogorov equations, okay, in the continuum limit. So let's do it. And again, if you don't know it, it's not a problem. Everything is self-contained here. So are you OK with the definitions of the transition probabilities and with the definitions of the xi? Because we're going to probably spend the rest of the day with them. So make sure you understand this, the definitions. If not, stop me. Everything clear? OK. So x0 is equal to what? 0. Okay. Once we're at 0, it's also an absorbing state, so we're there. Xn is equal to 1. That we wrote here. So these are the simple stuff. And now the interesting thing is for I going from 1 to n minus 1, we can write the following. Xi is the probability to fix starting at state I. Let's say I'm at state I, and I took one more step. Well, what are my possibilities? I can either stay at I, go to I minus 1, or go to I plus 1. Okay? This is everything. So I can write that the probability to fix, given that I started at state I, is the probability that I went from I to I minus 1, that I already have here. So pi I minus 1. And then given that I'm at state I minus 1, what is my probability to fix? By definition, xi minus 1. But that's not the only possibility. There is a chance that I went from I to I plus 1. And then what's my probability to fix? xi plus 1. And then there is, of course, the probability to stay at I times xi. So now I have enough equations. This determines xi. These self-consistent equations determine xi. Okay? And what we're going to do next is we'll solve this very easily. Okay? You already know the answer, so you're going to tell me in a second what it is without trying to solve it. You don't need any algebra to solve it because we know the answer. And then we'll do the more general case, which is not neutral. So just to check your understanding, where on the board did I assume neutral evolution? By now you know what neutral evolution is. So where is the assumption of neutrality here? Exactly. In this equation here, where I assume that I'm choosing the cells for reproduction equally for the A and B types. It's just their fraction in the population. So here and here is encoded the fact that it's neutral evolution. Now, for neutral evolution, we already said before, what is x1? Somebody said it before. It's the probability that one mutant fixes. Very small probability. What is it? 1 by n. So what do you think xi is? If I start with I individuals of A, what is the chance that one of them, they're all identical, so what is the chance that one of them overtakes the population? I over n. So you can plug this into this and see that it indeed solves it for these particular probabilities. Okay, so here it was just a warm-up. It's not interesting because we know the answer without any calculations. This is neutral. So now we're going to go beyond neutral. So what do we have to do? We have to modify these probabilities. This equation is still correct because it's completely general. The logic behind it is just saying I can go up in the ladder of states, I can go down in the ladder of states or stay, and it has, so this is still fine. Okay? Everything here actually is still fine. The only thing we need to modify are these equations, and then we need to solve the equation. So that's what we're going to do next. Any questions? Okay. So, running out of chalk here. I'm going to erase this, because this is no longer going to be true. And I'm going to erase this. And I'm going to define that the transition P from I to I minus 1, I'm going to be completely generic, and I'm going to call that beta I. Later, we'll substitute particular forms for beta I, corresponding to a specific model. But for now, it's general. This is going to call beta I, and Pi I plus 1, we're going to call alpha I's. Okay? And now, the equation that we have here takes the form x naught is equal to 0, xn is equal to 1, and xi is equal to beta I x minus I minus 1 plus 1 minus alpha I minus beta I xi plus alpha I xi plus 1. So this is the most important equation. It's the recursive equation where I use this general form for going up and down that will take into account the asymmetry in the growth rates between the two species. Okay? So that's what we're going to solve. And now comes a trick. And the trick is to introduce a new variable that we're going to call yi, where yi is xi minus xi minus 1. So we're looking at the difference. Okay, remember, xi's are these fixation probabilities starting with state I. Now we have a formal problem. We define the problem completely. And to solve it, it's an algebraic trick of looking at the differences between xi and xi minus 1. Okay, why? Because we're going to get some magic and some nice cancellations here. Okay, so I can't give you, better intuition for why I'm making this step. But these equations come up a lot, right? So in general, this sort of equation is called the first passage time equation. Okay, what is the first passage time? I have a stochastic process, very similar to a random walk. Okay, it's not exact. We're going to talk about random walks probably tomorrow. But if you think about the number of cells of type A, then it can go up, it can go down, and it's a stochastic process. So it's some kind of a non-conventional random walk. And we stop the process when we get either to 0, we didn't fix, or n, we fixed. Okay? So this sort of problem is called the first passage time. And there are a lot of problems that essentially map mathematically into problems of this type. So in fact, a year and a half ago, I was teaching in a summer school in Japan, a kind of a slightly longer course than here on biophysics. And I was talking about this problem and fixation probabilities. And then I realized that we are working on a problem that has nothing to do with evolution. It's a problem about chromosome segregation, okay, of human cells, okay, where there were 46 chromosomes, and each one of them are segregated into one daughter cell or the other daughter cells. And we were interested to know how long until all the chromosomes are correctly segregated, because sometimes you put 2 chromosomes into one daughter cell, and then the process will not divide, because if you'll divide at that stage, you will die. Okay? So it's a process where you have chromosomes becoming correct or incorrect. Okay? So you can think about it as 46 pairs of chromosomes, each doing transitions from a correct state to an incorrect state. But that's not a good way to think about it. And a better way to think about it is to say how many correct chromosomes I have. And if you call that I, then you get very similar equations to this. If I have I correct chromosomes, I can either become, in the next point, I plus 1 or I minus 1. And mathematically, the problem looks very similar, not identical, but similar to this one. Okay? And it's also a first passage time problem, because the question we ask is how many steps do I need to do until all my chromosomes are in the correct state, until I get essentially to the end state. Okay? And this trick that we're doing here was, I realized when I was teaching, it's exactly what we needed for that problem as well, and it helped us make an advance of that problem of chromosome segregation. Okay, so sometimes the same math appears in all sorts of places, even though if the biology or the physics are quite different. The application can be quite different. Okay, so let's see what this gives us. So I plug in this yi, and I define this yi, and now we have y1 is equal to simply to x1 because x0 is 0. So that's one result we have. We have that the sum of yi's, I goes from 1 to n, is simply equal to x1 minus x0, which is 0, plus x2 minus x1, plus x3 minus x2. So then we have these telescopic sums that cancels. And in the end, we find that this is simply equal to xn, which is equal to 1. So this will also be useful to know that the sum of all the yi's simply sum up to 1. Is this clear? Yes? No? Okay. So far it's been algebra. But now, okay, I need some more chalk. Maybe there is over there. Yes. So now we need, the most important thing is if we use this equation. So I leave it to you as an exercise, and please try to do it because I think you'll learn it much better if you do it yourself. Simple algebra, if you use this equation, the defining characteristic of the recursive relation we got for the xi, and you use this definition of the yi's, and I think it's much better if you do it yourself, you will find that basically yi plus 1 is equal to beta I over alpha I times yi. And that's the magic. Okay, so by kind of looking at these differences, instead of this messy recursive equation that involves xi minus 1 and xi plus 1, we get this very simple relation between yi plus 1 and yi. And we can call beta I over alpha I gamma I. Okay, if anybody has trouble in deriving this, then please come to me before or after tomorrow's class, and we can do this together. Okay, but it's just simple algebra. And now you understand that we're basically done, right? Gamma I is something we know. Remember that beta I and alpha I are not variables. They're part of the definition of the problem. They're telling you your rates of going up and down in the state ladder. And now we know that y1 is equal to x1. So we know that y2 is equal to gamma 1 times x1. And y3 is equal to gamma 1, gamma 2 times x1. And so on and so forth until yn is equal to gamma 1, gamma 2, gamma n minus 1 x1. Okay? And then so if I have all of these formulas for the y's, what else do I need to do to find x1? What's the last step? What's that? Exactly. So we had this sum of yi's is equal to 1. Okay? And that will give me a formula for x1. So we'll find that x1, 1 plus gamma 1 plus gamma 1, gamma 2 plus gamma 1, gamma 2 to gamma n minus 1, all of that is equal to 1. And so we're done. We find that x1 is simply equal to 1 over 1 plus a sum over the product. Sum goes from 1 to n minus 1 of the product of the gamma k's from 1 to, let's say the index here is j, from 1 to j. Okay, but it's basically this sum here. So we get a messy but explicit formula for the fixation probability. It's still hard to see the physics behind it, and that's what we're going to do next. So now we're going to define the alpha i's and the beta i's, plug it into this very general formula to understand what I told you before. Why is, if you have a fitness advantage of 10%, you have a fixation probability of 10% or something proportional to that. So that was the key result we wanted to prove, and I'm going to show you how it pops out of this nice formula here. Okay? So again, zooming out, we're studying a Markov chain characterized by these alpha i's and beta i's. We looked at the fixation probability starting from state I. We call that xi. It obeyed this recursive equation. We did an algebraic trick of looking at the differences, and that allowed us to solve the recursive equation and obtain an explicit formula for the fixation probabilities. Okay, that's what we did. And now we're just going to analyze it in slightly more detail. Okay, so let's look at the case where the probability to choose A is given by Ri divided by Ri plus n minus I. Okay, so let's stare at it for a second and understand why that's a reasonable model for evolution, for selection, okay, for the fitness advantage. If R is equal to 1, this is just what we had before. The chance of choosing A is I divided by n. It's the proportion. Okay? But if I have R larger than 1, then I'm biasing my chance of selecting selecting A species to be Ri instead of I. So this is like a partition function in a standard stat mech, right? This is my Ri divided by Ri plus the chance of choosing the B. And if R is smaller than 1, then I'm biasing against selecting the A types. Is this clear? Okay, so this is my chance of selecting the I, okay? And the probability of eliminating the B is the same, right? We said that for the elimination, it's still just by proportion. So in this case, Pi minus 1 is simply to go down, okay? I need n minus I. That's the chance of selecting the B types divided by what? Excellent. Ri plus n minus I. Okay? This is my chance of selecting the B types. This is my chance of selecting the A types. They sum up to 1. And all of that is multiplied by the chance of kicking out the A types, which is simply I over n. Okay? So that's my chance of going down the ladder. And my chance of going up the ladder by the same rationale is I need to reproduce A, which is Ri divided by Ri plus n minus I, and kick out a B. What's the chance of kicking out a B? Perfect. N minus I over n. No selection here. All the selection is in the growth rate advantage. So now that defines my alphas and betas, and gamma I was the ratio of the beta i's going down the ladder divided by alpha i's. And this is simply Pi I minus 1 divided by Pi I plus 1. So all I need to do is take this and divide it by this. And you can see that a lot of terms are common to them, the n minus i's and the i's. And when you do that, you find that this is equal to 1 over r. So we get a very simple result that all these gammas that you would have thought depend on the index I explicitly, for this particular choice, become simply a constant, 1 over r. And now we can go back to the formula, plug this into this formula here, okay? And we have to do a little sum over a geometric series, because this is simply 1 over r to the j, which again I leave to you as an exercise. And that gives us this very elegant formula that the probability of fixation is equal to 1 minus 1 over r divided by 1 minus 1 over r to the n. Yes? Here? Where, here? Ri divided by r times I. Oh, the dot was missing? This is what? Okay, yes, it's r times I. Okay? So again, I'm biasing. Instead of just being I give it a bias of, say, 10% if r is 1.1. Everybody on board? Okay, yes? Good. So the answer is yes and no. So the results are completely generic. Like I said, you can take different models for evolution, like Wright-Fisher, like serial dilution. So if the notes I uploaded, I do the calculation for a serial dilution. And you basically get the details will be different. But if you take r close to 1, being typically when you have beneficial mutations in nature, they're not going to be factor of 3. They're going to be mild effects. Most mutations in nature are deleterious. The ones that are beneficial are typically mildly beneficial. And if you take the different models for mildly beneficial regime, they're all giving you the same results, right? Fisher, serial dilution, Moran process. So there the details don't matter.
So should I see R be lambda A divided by lambda B?
Yes, yes. So you can think exactly. You can think of R in the language we had before as lambda 1 divided by lambda 2, and it's going to be approximately equal than 1. Okay, so the two growth rates are pretty close to each other. Lambda 1 is slightly better than lambda 2. And if we call that fitness advantage epsilon, so if we call r minus 1 epsilon and assume it's small, we can take this formula, okay, and expand it. And you will see that all of this, okay, the r to the n we can forget about for a large population. It's going to be negligible, so we can forget about this term. Okay? And this will be approximately 1 minus 1 over 1 plus epsilon, which is approximately epsilon. And this is the result that I promised you, that if I have a small fitness advantage of a few percent, that few percent becomes the fixation probability. And that's a very robust result that's independent of the model. Okay, so this is an important result. By the way, another limit you can try and take it as a third exercise, if you want to understand things better. If you take the limit of r approaching 1, being neutral evolution, but for a fixed n, what does all of this thing must become? 1 over n, because that's the neutral case we understood before. So this formula has the advantage that it captures both the neutral evolution and also the more interesting case of beneficial mutations. Another thing you can do is if you take r to be smaller than 1 and now take n to be large, the probability of fixation becomes 0. You cannot fix in this case. Okay, so this general formula is quite elegant and captures a lot of scenarios. Okay. Yes.
This is equal to x1 prefix.
What's the question?
Is prefix,
Yes, yes, yes. Prefix is what I called before x1. Absolutely. Good. Okay, but it's what we were after. So we had like all these xi's, again, because we needed to solve for all the x's. Now we also know the probability of x7. It was just easier for us than to solve just for x1. Okay, but that was the quantity we're after. So the result is quite remarkable. No matter, even if I take a huge population of 10 to the 8 cells, the probability of fixing a 10% advantage doesn't depend on n. N doesn't appear in this result in the limit of a large n. I don't think it's obvious because, like,
but it's obvious that if I make
So I'm not aware of a simple argument for that. If you have one, I'd love to hear. Because, like, the point is this. I have a large population. Initially, it does this random walk. When it gets to some certain threshold, it overtakes the population and everything becomes deterministic. Why would this behavior cancel, like not involve n?
No, it's not like.
Let's discuss it.
It's surprising, but mathematically, once I get to the result and study the large n limit, I'm kind of expecting that it doesn't depend on n if I'm studying the limit for n goes to infinity.
But you could have been in the case where the result does depend on n even at the end. It happens to be not the case, but I'm not aware of any kind of.
No, it's just the mathematics.
No, so I asked actually many people, like, can you give me simple intuition for that? And I wasn't able to come with a kind of simple, transparent argument for why it has to be that way, that the result is independent of n. So I personally kind of find it somewhat surprising. One last thing I'm going to say before we conclude for today is actually, let's talk, and it's related to this comment about n. Okay? So let's go to the opposite case. So we understand that, if you think about the Lenski experiment, n is huge. N is 10 to the 6 to 10 to the 8. So we are in this large n regime. I have a beneficial mutation. It happened. Maybe it fixes. Maybe it doesn't. Okay? And we only see in the experiment the stuff that fixed. We suddenly see a new mutation overtaking the population. It's called the genetic sweep. And because we have everything in the freezers, you can take it out and really see, look, after 15 years, this mutation arose in this point in the genome, and it overtook the entire population in a short time. Okay? So we can see these genetic sweeps. But let's look at the opposite regime, Gedanken experiment. Let's assume that evolution is neutral. If evolution is neutral, the probability of fixing any mutation is 1 over n. Right? Now, let's say I have n individuals. The rate of fixing new mutations is 1 over n for each mutation times the mutation rate, let's call it mu, like we understood in Luria-Delbruck was a way to measure the mutation rates from the Luria-Delbruck experiment. So mu is, 10 to the minus 8 or something, 10 to the minus 9. 1 over n is the probability of fixing a mutation. But how many individuals do I have? N. So now again, n cancels, but for a different reason. This is the rate of fixing new mutations in my population, and it's independent of the population size. So that means that if I have two populations of, bacteria or rabbits or whatever, and I look how many mutations they're different from each other, I can tell you how far in the past they separated from each other. So this is called the molecular clock. Because this is independent of n, and mu is a property of error corrections in the genome and kind of, something that's more or less hard-coded into the biology, I can use the number of mutations as a way to measure how much time has passed. So this idea is called the molecular clock, but it hinges on the assumption that evolution is neutral. Now, for the most part, most mutations will have a small effect, but we see in the Lenski experiments that beneficial mutations are important in driving evolution. So, okay, it's not obvious that you can always use this assumption of neutrality. Okay, so we're going to stop here, and tomorrow we're going to, now that we understand that fixation is proportional to the fitness, we can ask, can we understand this fitness trajectory and within a simple model? And the answer will be no. We're going to kind of discuss a model, and I'll show you that things are not consistent with a slow increase of the fitness. You expect a faster increase to a plateau within the simplest model of evolution. And that will conclude our discussion of evolution. And then, mathematically, we're going to stay in the same regime of stochasticity and random walks, but the details will be different, and we're going to talk about cells size control. So it's also related to E. coli growth and divisions, but different aspects of it. Not evolution, but now zooming in to the property of a single cell and how it controls its size. Okay, so I'll see you tomorrow. And make sure you signed your name here.
Yes, before you, just a small announcement. So the next week we are going to have this dinner, social dinner, the last supper offered by Gestat. And it's going to be on Thursday, 13. But you have to write here whether you will come or not, okay? Because we have to give a precise number, okay? So it's another.
To know if I get everything.